I appreciate the opportunity to be here and share with you a little bit about NeoSTEM's progress on one of our lead programs in cardiovascular disease. And like so many of you, we're focused on trying to use cell therapy to cure disease as opposed to treating symptoms and manufacture with highest quality and lowest cost. So NeoSTEM is a public company. We're on the NASDAQ exchange. And just to give you a little bit of idea, we have $41 million in the bank, so we can hit some of the milestones we have ahead on our clinical programs. We have headquarters in New York with contract manufacturing facilities in Allendale, New Jersey, Mountain View, California through PCT, as well as a new facility which we've added through the merger or the purchase of California Stem Cell in Irvine, California. So we've now added to the NeoSTEM family. We have 155 employees, and I thought the best way to describe who we are is to think of us as this contract manufacturing organization that works on manufacturing, development, automation, engineering, ways to lower cost of goods, produce high-quality products that we can do for clients and for those in the regenerative medicine space who want to outsource the contract manufacturing, as well as allow us to internally develop our own pipeline products. And we're in 4 major areas: cancer, autoimmune disorders, tissue regeneration, and cardiovascular disease. And so I thought today I would spend my time really talking about our CD34 program, which is focused on ischemic repair. We did just buy, as I said, California Stem Cell, which is a dendritic vaccine that focuses on a person's autologous tumor, so we actually target the replicating cells and the stem cells that are causing the cancer. We have very exciting phase 2 data with melanoma, and I think Bob's going to talk a little bit about that on Thursday, as well as our immune modulation, which is in partnership with Becton Dickinson, and we'll be focusing on type 1 diabetes, which we'll see data from Jeff Bluestone in June at the ADA meeting. So those are some of our programs, but let's talk a little bit about cardiovascular disease. So I know from Juventus you heard a little bit, so I won't bore you with some of the things you've already heard about CD34 cells and the way they migrate along the STF1 gradient. But the idea is, how do we use our natural body's repair mechanism to heal after an infarct? So as you can see from this heart, the bottom part and the apex is the infarct zone. And what happens is, normally, your body, when it becomes infarcted and doesn't get oxygen, sends a message to the bone marrow for the CD34 cells to leave the bone marrow, go to the area of injury, create a paracrine effect, angiogenesis, and really lay down blood vessels and bring oxygen to the area. But this doesn't happen effectively from that cardiac signal in many people. So about 20% of people with a STEMI do not have that kind of response. And you get apoptosis and cardiac myocyte death. And what ends up happening is there really hasn't been, even with standard of care, changes of how people fare once they survive a heart attack within those first 30 days. So the idea is to come up with a way to give them their own CD34 cells, the ones that migrate, the CXCR4 cells, deliver them into the coronary artery, which will then hone to the area of injury and lay down the blood vessels to preserve that heart muscle function. Because if not, you end up with what we've seen here, which is, ventricular remodeling. And so you have to look at different ways. One is perfusion, the other is function. And so what can you do to preserve the heart muscle to be able to pump for the activity that it's needed and not get worse with time? So we've seen a lot of work over the last 10 to 15 years, a lot of meta-analysis using CD34 cells to treat cardiovascular disease and post-MI. And the one theme that we see in common is, if you give enough cells, it's shown to have improvement in function and event-free survival. So that's what we've seen as the theme of all these meta-analyses put together, is that there definitely is a signal, and we're excited about our product, AMR-001. So the idea is how do we preserve that heart muscle function so that you don't get repeat heart attacks, arrhythmias, premature death, which you can see from that boggy heart on the right, versus shrinking that infarct size and bringing oxygen to the area to preserve that muscle. So NeoSTEM has created a therapy called AMR-001, and the idea is a person comes in, has a heart attack, gets angiography, gets stented, and at day number 4, if their ejection fraction hasn't recovered, they're eligible for the therapy. We do a bone marrow aspirate, send the cells to our lab, pull out the CD34 CXCR4 cells, stabilize them, send them back to the doctor. It's a 72-hour shelf life, and those cells are infused right through the stent and through an angiocath 5 to 10 days after the infarct. And that's the time when the SDF1 gradient is the highest, that those cells will then mobilize to the area and lay down the blood vessels and have an effect on the area. So the question is, do cells actually react to a person that has a STEMI? And what we've seen is that post-STEMI, within about 12 to 24 hours, people's levels of CD34, CD45 positive, Lin negative cells go up. So it happens, it's repairing, and there are a portion of people that will have cardiac stenting and be fine and not be really have the sequelae of this horrible STEMI. But the 20% that do progressively get worse, and if you're age 45 and over, you have a 1 in 5 chance dying within a year. So do the cells actually know to go where they're needed? Well, what we've seen here is that there is outcome data post-MI that shows that patients that are able to mobilize the CD34 cells have the best clinical outcomes. And so we've seen a couple of articles suggesting this, and that is further supported by Dr. Zaire's work, which we saw in the European Journal about a month or two ago, where he actually showed the relationship between CD34 that are SDF1 mobile and event-free survival. So you see at 6 months, the curves start separating, and it's sustainable at 5 years. And he showed that the migratory capacity of these cells is associated post-MI with 5-year event-free survival. So it's pretty exciting data, and our product is based on these CD34 CXCR4 cells and the way they migrate to the area of injury to lay down new blood vessels. So we have method and composition of matter. We have very strong IP, and we believe that this is a therapy that could make a big difference in treating a patient after they have a heart attack. So do they impact perfusion? Well, we showed in our phase 1 study that yes, if you had enough cells, and that's the key, you have to have enough cells to get the imparted benefit that you're looking for. And we showed that if a patient received 10 million cells or more, they had an improvement in perfusion. And that's the negative scores on the bottom. It's like golf, the more negative, the better. And we can see that those people did better. We also showed that with a relationship to the SDF1 mobile cells, the CXCR4, CD34 cells, there was a decrease in the infarct size, as well as the perfusion, which we saw on the last slide. And so that improved with an increase in those CD34 mobile cells. And ultimately, what we care about is function. So it's great to bring oxygen to the area. It's great to decrease the diminished part of the heart that is affected by the heart attack, but will it affect perfusion? And will it affect ejection fraction, which is function? And the answer is what we found in our phase 1 study, and it was 31 patients, but pretty interesting, not one single patient, not one, who received 10 million cells or more at 6 months had a decrease in their ejection fraction. So if it was in the control group or the 5 million cell group, they 30% and 40% respectively were worse at 6 months. So this was pretty impressive data. Overall, the cohort did improve, but in small numbers, and we're really looking forward to seeing if this, how this will show out when we do 160 patients in our phase 2 trial. So I think what's important here is to understand that we are just amplifying the body's natural repair mechanism, and that you don't have the risk of any genetic manipulation. They're autologous, so you don't have any immunogenicity issues, and that you actually deliver the cells where they're needed in high concentrations. And because they're your own cells, you're not manipulating them. We've been shown that they're safe. We've seen many people get CD34 cells, both for bone marrow transplantation, as well as a lot of the studies that we've seen over the last 10 or so years in the heart. And so it's deemed, pretty much to be safe and effective. And so to really look at that effectiveness, we did a phase 2 study, 160 patients, double-blinded, randomized control. Half got 10 million cells or more, and the other half got media. And what we did is we used perfusion, because that's the mechanism of action for the primary endpoint, and then we measured all other measures of function, so ejection fraction, systolic volume, wall motion function, quality of life scores. We'll also be following for 3 years MACE to look at adverse cardiac events. And we are expecting the data from this trial. The last patient was enrolled in December, so the last patient that was enrolled will have their data, 6-month data, in June, and we'll be able to close the data set in the end of this year. Before that time, before AHA or at AHA, we'll be giving those results. So the question is it just an acute MI that these cells can have an effect? And we weren't really sure until we saw this interesting data that was done in Slovenia. 110 patients were treated, and what was different is they weren't injecting these cells into the heart muscle. These were actually put into the coronary arteries, and they mobilized to the area of injury. And actually at 5 years, they showed for chronic heart failure, there was improvements in survival, in decrease in cardiac events, heart failure, and to heart transplants. So this was a pretty interesting study, and I think what was most important to us, which is why we were a little hesitant to go out there, is we didn't want to be limited by a NOGA mapping system because not everyone has it. So this way, all you need is a cardiac cath lab, so your distribution will be a lot greater. So we're pretty excited about these results. I mean, it's 5-year results, which is pretty incredible. So it was one center, but we'll be starting a phase 1b/2a trial in congestive heart failure in the balance of this year. Again, for all of us, as we develop cell therapy products, we think intellectual property is very important, and we're very focused on having both method, composition of matter, both in the United States and globally. And our hope will be that we'll be able to partner, once we hopefully get good data for our phase 2 trial, we'll be able to partner some geographic locations. We have other indications that we can go into, and this is one of our platform technologies, and obviously we're interested in big markets like congestive heart failure with 23 million people with congestive heart failure. Other areas could include traumatic brain injury, chronic limb ischemia. So we're really excited about this acquisition. We bought the company in October of 2011. We've done the phase 2 trial, and we'll have the data read out the end of this year. So we've been very efficient in taking a phase 1 asset, taking it through to phase 2, and preparing for phase 3, hopefully with good data. We have a pretty exciting scientific advisory board who helps us look at other clinical indications where we think the therapy may have benefit, and we'll continue to look for both partnerships and other areas to use AMR01. So in summary, this is an autologous CD34 bone marrow-derived stem cell program for interventional cardiologists that, the key is you can get perfusion where it's needed, you can affect ventricular function, these cells go to where they're needed, they're autologous and safe, and we believe this can be a reliable pharmacological product in the future, and are hoping for exciting results and to continue development to get to commercialization as quickly as we can. Any questions?
Well done, Robin. Yeah, we've got time for some questions.
Yeah.
Well done. Very exciting. You are so busy, it is untrue, but making great progress. We've got some questions for Robin. Robin, I'll just kick off. I mean, clearly a lot of different platforms, a lot of different technologies, and you've got manufacturing, which you've integrated in, and I like that very much. So how do you see it? Do you see it that your companies will be the primary source user of the manufacturing, or do you see very much the manufacturing being really supplying the industry and you'll have a small part of it? And how do you see the balance there between the two?
We see that the manufacturing business supplies the industry. I mean, it's a leader in the development and manufacturing and looking for automation engineering solutions to help lower cost of goods. We just benefit as being a part of it, but we keep the two businesses very separate. And of course, 15 years of experience in developing cell therapies is helpful for our own internal programs. But as you said, we're in the CD34 space. We have, the dendritic vaccine. We have the Treg program for autoimmunity, and then our V cells for regenerative medicine. So we're in a lot of different cell types, and we'll be continuing to develop. The goal for us is to get products to market as quickly as we can to treat chronic disease.
The California stem cell one was a major coup, I think, wasn't it? I mean, that was quite a
We're pretty
wave.
excited about that.
And I agree. Could you just say a little bit more about that program?
Sure. So they had amazing data. So basically, the theory is that when you're treating cancer now, you're not really just treating cancer cells to kill them. You want to make sure that you're killing the initiating cells or the stem cells that will cause metastatic disease. So the idea is to take the person's tumor, their own personalized tumor with their own genetic mutations, and make a vaccine that makes the immune system want to kill those cells. And so they worked on malignant melanoma.
This was Hans Kirstein's group.
Yes, and they were able to improve the manufacturing process. It was Dr. Dillman's work, and the data was incredible. 72% of the people who were treated in the therapeutic group had a survival at 2 years, which for melanoma is unbelievable, malignant melanoma. So we'll be starting a phase 3 trial. The FDA has given a SPA, a fast track, and orphan drug designation, and it's a small trial, 250 patients, and we're hoping to get it enrolled as quickly as we can. And
because this really gave you a very strong bit of pipeline. It gave you that pivotal clinical study, which you didn't have up to that stage. And so that gave you really a full pipeline of just about everything in the development thing. I mean, running that must be a real challenge. You've got everything going. I mean, prioritization, I mean, how do you balance everything?
That's a great question. I mean, you always have to prioritize. I think we prioritize later stage technologies, and that will help then fund some of the earlier stage. We just entered into a licensing deal for China for the liver, and so we'll continue to partner and reprioritize. I think, you know, the contract manufacturing business is there to help us on the manufacturing side, and we have a great clinical team with experts in each of the fields to help move these along. We'll work with strong CROs, you know, to get the trials enrolled and you know, get to data.
Very exciting. Any other quick questions for Robin? I can't. Okay, fine. If not, so can we thank Robin for a great
Thank you.
great talk.